Who? Me? What about? I don't know. Just a grin. Oh, multiple test. Right. Thank you. Hey? Do I get cold? I get cold on the top, not on the bottom. Yeah. I wear jumpers all the time, but not long pants. Yeah. That's it. Woolen blue one. Yeah. Oh. It could have been worse. You could have said pop jumper. I'd have smacked you for that. All right. Today is all about yeasts. So it's kind of a bit easier than moulds because they're really identified by technology as opposed to by microscopy. So it's all about yeasts. And the first one is a particular yeast called Coccus formans, and this yeast has got a sexual state and an asexual state. And when it's reproducing sexually, we call it Phylobasidiella because it's a basidiomycete, and the basidiomycete group of fungi are actually mushrooms. So this is a yeast that can exist kind of like a mushroom in its other sexual state. A bit confusing, but don't worry about it for now. What we're keen on is this organism as a yeast. But to some degree, this bug as a mushroom type organism is also important in part of the story. As a yeast, there are 4 different serotypes. It's got a capsule, and you can serotype the capsule. And some time ago, they separated the yeast into 2 varieties. So C. var neoformans, so the variety is neoformans, and the other one is variety gattii. It was important because gattii tended to cause a different kind of disease in different people than var neoformans. And then to make things more complicated, they decided to change the name. So the var gattii was renamed not as a variety of C. neoformans, but as a whole different species. So Bacillus sporus is the new name for C. neoformans var gattii. I only labour this point is when you read stuff, you read more than one name, and you've got to understand that if you read C. Bacillus sporus, it's really referring to what was once called C. neoformans var gattii, and it really is important in the sense that they cause different kinds of diseases. And they also renamed one of these varieties as var grubii, that's different serotypes. So to summarise it all, so it's not too confusing, there used to be C. neoformans, var neoformans, 2 of those, different serotypes. They've renamed one a separate variety. This is less important, this change from here to here, than the change from down here. So what was C. neoformans var gattii is now just C. Bacillus sporus. I use the term var gattii a lot because to me historically it's still the term I'm more familiar with. The reason it's important is C. Bacillus sporus, what was once called var gattii, causes disease more often in immunocompetent people. Whereas C. neoformans var neoformans and C. neoformans var grubii, the top ones, are more often linked to HIV positive people, especially var grubii. Hope that's not too confusing. It is really annoying. I think out of all the parts of micro, mycology is the one that's undergone the most changes in nomenclature in the last 20 years. It's really quite irritating, but that's the way it is. So var gattii or Bacillus sporus, more often in immunocompetent Bargrubii is more common if you're HIV positive. And this really is a yeast that's all about HIV, certainly in my experience it is. So there are some unique ways in which this lives in the environment. So C-neoformans, the one that causes disease more often in HIV positive patients, has a strong link to bird excreta. You find it around bird droppings historically. So you'd see it occurring in people that handle birds, people that worked in old buildings where there were bird droppings in the ceiling of the buildings. C. bacillosporus, what was once called Gattii, has a strong link to a particular kind of gum tree called Eucalyptus camaldulensis. This is the river red gum. A bit more to that story as well, which we'll come to in a minute. It seems a bit odd that one's linked to birds and one that's linked to trees. There's kind of a bit of a disconnect there. In the end, probably this one that's linked to birds has also a link to a certain tree. They just haven't found it yet. And this one linked to trees possibly has an animal that's linked to these trees. So it's a tree-animal-human connection. And what we know is that there are a couple of enzymes that are important in the organism that kind of fit with where it lives in its environment, its habitat. So cryptococci in general are urease positive, and that makes sense if you spend your time living in a high urea environment, and bird poo is high in urea. So being urease positive means you can cope with that environment. You can break down the urea. Makes sense. Phenyloxidase is an important enzyme for the organism, both in terms of its identification and also in terms of what it does in your body and where it lives. And phenyloxidase is useful in breaking down phenol compounds, and phenol compounds are very, very strong in the bark of a whole range of trees. It seems that it's got these enzymes that do a number of things: help it survive in the environment, also help us identify it, and also help it live in your body when it's doing that as a spare part job. So it's a major part of its life is in the environment. It causes human disease really by accident. This is kind of how the story sort of works. There's probably a host plant, and we know that for Bargrubii, or the new name Bacillosporus, host plant is a gum tree. And there's actually a range of other trees now besides just that one gum tree, but that one gum tree was the first one that was discovered in Australia. And the host plant for the other species or other variety is unknown. When it lives on the host plant, it's living as a sexually reproducing basidiomycete. In other words, it's living as a mushroom type organism. It doesn't live physically like a mushroom, but its fungal structure and the way it reproduces is like fungi do, but it's not actually physically a fungus, a mushroom. And when it's growing in that state, it can release basidiospores. And we've also seen that there's often a spike in infection at the time that plants flower. So maybe when the plants are flowering, these fungi are also reproducing and leaving out spores. Now, a number of things happen. Possibly the basidiospores, the sexual spores, can mature and change into yeast cells for the capsule. That's one possibility. The other possibility is that the basidiospores infect animals, birds for neoformans and koalas for Bacillosporus. And it's maybe in those animals where the change from basidiomycete sexual state to the asexual cryptococcus state occurs. And then in the dung of these animals, either bird droppings or koala dung, then they're excreting yeast with capsules that then become dried and we inhale them. You can get them from birds when they're in the bird droppings, or we tend to get them from around the habitat of the trees in which the koalas live. Now, there's got to be more to this than koala bears because, I'll explain this. One of the reasons why I looked really hard at this for this gum tree is I found a whole range of infections with Vargatii occurring on the east coast of Australia. And they try to find some particular thing in the environment that matched that distribution. And they discovered that gum trees, this particular gum tree, was most common on that part of Australia. And they went out and searched, spent a couple of years taking samples around gum trees to find the first ever positive sample. They've since found that this gum tree has been exported to other parts of the world, California in particular, who also have problems with Vargatii. So there's a link between this gum tree and infection. We've now found a whole range of other trees that also can be linked to infection. So the link to the tree is quite strong. But where these trees have been exported, there's no koalas. Must be a second animal somewhere in this whole complex story. Doesn't matter for us. In the end, we don't care where we get it from. We just care that we've got it. So the story in the background is quite interesting, but it's a bit complicated. What do we see as disease? It really, as a pathogen, is opportunistic. It can cause disease if you're immunocompetent, but even then requires some kind of major exposure. It's not an aggressive pathogen, but it's much more common if you're immunocompromised, and it's especially been linked to HIV. So the rise in this organism being detected and diagnosed almost parallels the discovery of the HIV virus and AIDS and the AIDS epidemic. It can either be a chronic disease or a subacute disease. It's rarely ever an acute disease. It's not as though you're exposed today, sick tomorrow, dead the next day. You're exposed today, you might get infected, you might not, bless you. If you're infected, it's a slow disease, it's indolent, ongoing, it's not really super aggressive. And what's amazing is it can survive in your body in huge numbers, and you make a very, very dampened immune response. It can almost keep growing you ad nauseum for a long time, become a huge part of your body mass, and you're still alive. We know that infection is acquired by the respiratory route, so you breathe them in. And here's part of the problem is that when you look at the mechanics of breathing things in, the size of the yeast cell with a capsule is too big to be breathed in to get as far as your lung. So we either breathe in a yeast form that's desiccated, in other words, it's dried up and got smaller, or in actual fact we're breathing in basidius spores of the sexual state, and they transform into the asexual state in your body. And no one knows which of those two is true yet. You do surveys in populations where these organisms are common, there's quite a reasonably high seropositivity rate. So lots of people are antibody positive to Cryptococcus, but they've never had any disease. What that means is most often, if it's respiratory, it's infecting you, you make an immune response, but you're asymptomatic. If you're unlucky, and that might be because you're HIV positive or you're otherwise immunocompromised or you're just dead unlucky, then it can most commonly cause disease in the central nervous system. And it's most commonly meningitis. If it's Vargatii, it often tends to cause a more focal disease that's not so spread out, and we call that a cryptococcoma. What that really means is, the oma part means tumour. It almost grows in your body site like a tumour. That can be in your lung or in your brain. So rather than being diffuse meningitis, it might be a brain abscess almost. And in the lung, rather than being diffuse pneumonia, there's actually a big chunk of organism in your lung. And in some people, it'll disseminate, and sometimes the first sign of disease will be a cutaneous lesion on your skin. You breathe it in, it's been respiratory, it's been asymptomatic, comes out in your skin. Or it may go to other organs, spleen, liver, lungs, bone marrow. So the bug's quite flexible in what it does. This is an example of a big lung abscess from Cryptococcus, and some of these pictures are a bit gruesome. This is a disease from Australia. This is an Aboriginal man who survived, but he's had his lungs taken out to survive. This is Vargatii, and all this white stuff there is tumour. Tumour caused, but it's not tumour in the sense, it's not his cells, it's a tumour-like growth of the yeast. I think it's only one lung. I'm not great, yeah, otherwise he'd be out of breath, wouldn't he? It's only one lung, but it's just laid right out. This is all organism. Now, the idea you can be alive at the point this lung is taken out, have that much organism in you, tells you just how much you don't respond to the organism. It just seems to be in there like you're a big agar plate and just grows. This is one of my favourite all-time photographs. So there is the yeast cell, and there is the capsule. And this is somebody's spleen. And this person was walking around alive, HIV positive. But you would estimate this would be, what, a third or more of the mass of the spleen is just organism. If that was anything else, any other organ, or Staph aureus, you'd have been dead ages ago. I mean, Staph's only got to be a small percentage of your body mass and you're dead, whereas this can keep on growing. And most likely, the capsule protects the bug from being detected, and you don't make a huge immune response to the capsule. You make antibody to the capsule, and the antibody is not protective. So it can be antibody positive and still be reinfected. This is a primary skin lesion on HIV positive man. If you took that lesion and did a prep, that would be full of yeast. Primary skin lesion in somebody who was immunocompromised but not HIV positive. Skin lesion primary. Now, this is a cat. And there's the cat's eyes, and there's the cat's nose. That lesion, that lesion, that lesion, and that lesion, and that lesion are all yeast. And cats get a version of HIV. This is an HIV positive cat that's got infection with Cryptococcus. This organism is just, I love it. This is a porpoise that's got infection with Cryptococcus, and there's, I think that's the lung of the porpoise, and all these spots in here are organism. Now, how do you think that a porpoise gets infected with a yeast that's terrestrial? Is it terrestrial or terrestrial? Terrestrial. So in a minute I'll show you this. There's a big outbreak of infection. It's been occurring since 1995, I think. I just better check that number. In British Columbia. And there's lots of infection there, both in people and in porpoises. If I said to you, this is a graph of cases of AIDS, you would believe me, because it starts there in the late 70s, and we discovered it in the mid-80s, and it went like that. This is actually a graph showing the number of cases that diagnosed of infection with Cryptococcus. And it's a complete overlap and overlay of the AIDS epidemic. So initially, we saw this so much with HIV positive people. So about there, I was at university in this building studying hematology when we first saw the first paper clipping about HIV. So in my lifetime it was discovered and everything. It was amazing. I can remember sitting down there in that lab with my hematology lecturer talking about this thing in the paper about this AIDS thing. It was awesome. Long time ago. So Here we go. The most commonly diagnosed disease is meningitis. The most common disease would be pneumonia, but most often it's not diagnosed because it's asymptomatic. The most commonly diagnosed disease would be some kind of meningitis. You tend to see the immune response being lymphocytic, not being neutrophils. And there's no question it's strongly linked to HIV. So it's the most commonly life-threatening fungal infection in AIDS patients. It would surpass things like Candida albicans by a long way. And these days, I think back when it was first discovered, there wasn't as much emphasis there on preventing infection. You would get infected, then they'd deal with it. Now it's all a matter of preventing getting infection. So if you get disease now, it's a marker that you're being poorly managed. You shouldn't get this infection anymore because you should be able to manage your CD4 count, and when the CD4 count is low, they put you on prophylactic therapy that's antifungal to minimise the risk. So about 1 in 10, 1 in 5 people who's HIV positive are infected. Once you're positive, it's really hard to get rid of it. What you want to do is make sure you aren't infected. Once you're positive, removing it even with antifungal drugs is pretty difficult. And the issue there again is the enormous number of bugs that can be there, and you'll still be reasonably well. But once you've got that amount of organism, you're in a bit of strife. I wonder if they'll ever find the actual real mechanism for infection. So the debate again is about whether you inhale basidius spores, whether you inhale actual yeast. What's important though is the organism is not contagious between people, so you can't pass this on from individual to individual because the organism in your body is just too big to be breathed out as an aerosol, and then if you did, it would be too big to be breathed by somebody else. So it's not transmissible between people. The hallmark of the bug is the capsule, and when you see it in the lab in the pracs next week, it's a big gooey yeast, and sometimes it's so gooey if you leave the plates the right way up with the lids facing up and the agar facing down, it'll drip out into the bottom of the lid. The capsule is antiphagocytic. You make lots of it, so you often excrete the capsule in a whole range of body fluids, including urine. Quite possibly, the capsule interacts with the HIV virus, and there's been evidence showing you that the virus can gain better access to your white cells if there's capsule present than without the capsule. So maybe the capsule and the HIV virus somehow interact to aid in their access to the white cells. The bug makes phenyl oxidase. Now, dopamine, the chemical in your brain, is a form of a phenol compound, and phenyl oxidase assists the organism in metabolizing some of the chemicals that it finds in your brain. So that's why it likes your brain. The bug goes where it finds its best source of food, and because it's strongly phenyl oxidase positive and its habitat in the environment is bark of trees, somehow your brain and tree bark are quite similar chemically. Don't ask me how. It likes to go to your brain. So of your whole body, its preferred area is the brain. Phenyl oxidase is also an antioxidant, so it can reduce the capacity of your white cells to kill the organism. When you make an immune response, it's really important to have strong cell-mediated immunity, which is why HIV positive people suffer so badly. Do make antibody, but it's not protective. So we can use it as a screen to see about exposure in the population, but being antibody positive doesn't mean you won't get disease. Now let's cut to the chase. What we really want to know about is how do we recognise them, how do we identify them, and what do we do with them? Because the people can be relatively not sick, often you want to find this thing in a screening process. But if you have a specimen of spinal fluid, Doing a gram stain isn't always that useful, but an Indian ink stain is useful. Because the organism's got, it's reasonably smallish round yeast, so these yeasts are much more round than Candida, which is more oval. But it's got a capsule that's at least twice the diameter of the size of the cell. So the Indian ink stain is where you put Indian ink on a slide, add some spinal fluid, give it a mix, and the Indian ink will stain the actual middle part of the yeast, but not the capsule. So you end up with this slightly grey, round yeast with this great big clear halo. So it makes screening your spinal fluid reasonably good. The trouble is, as a test, it's very non-sensitive. About 40% of those specimens are culture positive, are negative by direct microscopy. So a positive Indian ink stain is great, it is infected, and it is a cryptococcus, but a negative one can't exclude the disease. We can do other kinds of tests. You can do other stains if it's a tissue biopsy, say from bone marrow or from lung or from spleen. You can do PAS stains or silver stains, and they work pretty well. Culturing, the bug grows easily on normal media, but you can't use a microbiotic plate. And why would you? If you had a specimen of spinal fluid and you cultured that on a microbiotic plate, why is that? It's just stupid. Sterile site. Culture sterile sites on media that grows everything and don't muck around with things that are selective. Same with chocolate agar. Don't use chocolate tracing, just use plain chocolate agar. This is a sterile site, and whatever grows is important, and the idea of having to have a plate that's selective is just dumb. So 37 degrees Celsius, it'll grow comfortably as a nice big shiny yeast colony in 24 to 48 hours. It's not slow at all. These grow like bacteria. Once you've grown one, then showing the capsule in vitro off your plate is really easy. Get a bit of colony onto a slide, bit of Indian ink, have a look, yeast, big halo. We'll do it in the prac next week. It's great. The urease positive. There are other yeasts that are urease positive, so it's not a great important test, but it is interesting to see them being urease positive. And we do, now this depends a lot, things are going to change I think. A primary plate that we use to identify the organism is a plate that contains phenyl oxidase. Sorry, a plate that can demonstrate the presence of phenyl oxidase. So one way of doing that historically was to use birdseed agar. And this is what it sounds like. You would go to the pet shop, you'd buy some birdseed, particular kind of birdseed, and you would put it in a blender, mash it all up, and the birdseed was very heavily loaded with caffeic acid. And caffeic acid is a substrate for phenyl oxidase and would go brown. On this agar, it would go brown. Because making birdseed agar was a bit complicated and you could never be sure whether one batch of birdseed was as good as the next batch of birdseed, it's easier to go to the shop and buy a bottle of caffeic acid and it works fine. And that way it's always the same amount of caffeic acid in your agar, which means your brown colour is reproducible. It's much safer doing things in a defined way than going and buying that. I've also seen people go in the bush, go and find a gum tree, get some bark of the gum tree and make agar out of gum tree bark and put the bark on there and it goes brown. So it works. I think this is just easier to do this. If you haven't got access to other technology, it's also possible to separate neoformans from Bacillosporus, the old Gattii. And the CGB agar is how this works. Cannabinine, glycine, bronchomyl blue agar. Cannabinine is slightly inhibitory. So, let me get this right in my head. The cannabinine inhibits var neoformans and allows var Gattii to grow. And then when var Gattii grows, it metabolises the glycine to make an alkaline pH change and it goes blue. So growth and blue equals var Gattii, or the new name Bacillosporus, have no growth and staying green equals micL4. But it won't separate the two separate serotypes in those two different varieties. You could, if you wanted to, go and buy some anti-sera and have anti-A, anti-B, anti-C and anti-D. Why wouldn't most labs do that? Do one every couple of years, and having all anti-sera in your fridge that goes out of date every 6 months to use for once every 4 years or something, not worth it. So this is pretty easy to make up, and we often make this up here and give it to the labs locally if they find one. If you have multi-top, then everything else is just forget about it. This has now become really, really useful. You may still use this plate as a screen, but multi-top is very good at separating, not only giving you the right identification, or separate them into the two different varieties. So it's become quite good. Not a great slide in the sense that this is much longer than they normally are because it's budding. They're normally quite round. Here's one that's just budding. So this is budded to that one. There's another bud there. But look at the size of the capsule. Big capsule. Now, when you think about capsules, I don't know about you, but to me, in my head, my mind says it's this round thing with a big lob of jelly all the way around it. When you look at the whole thing with a scanning electron microscope, what you find is the capsule's not a big lot of goo around it, but a whole series of projections. This is a capsule. These aren't fimbriae or flagella. These are polysaccharide extrusions. In actual fact, on microscopy, you think it's a whole thing, it's homogeneous, but it's not. It's a bit like this. Well, it doesn't matter, but it's just interesting. You form this idea in your head that it's going to be this big lot of goo, but it's not. Here's a plate. This is a birdseed agar plate or a caffeic acid plate, and this is a candidate albicans for comparison, and this is Coccus neoformans, and you see already that it's brown, but also you can see how wet it is. Wet, and it's got a capsule. And there's a CGB agar. It's a light green colour, and when they're positive, it goes blue. Note on this side here that you haven't done a streak plate, because the cannabinine is quite inhibitory. It's got to be a heavy inoculum, so you normally just do a lawn plate of your pure colony. Because microscopy is relatively poor, and because we often want to screen you for infection before you're infected, it's got to be a better way. So we can look for the polysaccharide capsule in your body fluids. So we aren't looking for antibody, looking for the capsule that you make in excess, or not you, the bug makes in excess. So we can get a reagent of latex with an antibody onto the capsule and do an agglutination and detect capsule in body fluids. Initially, it was done primarily to diagnose disease on those patients who were sick, who presented with illness. And if you do this test on CSF, it has at least a 90% sensitivity. So in other words, if you are infected and we do the test, it's almost always positive. If your disease disseminates, and it could be in your spleen or in your liver or somewhere else, could be in your lung, could be in your brain, if we take serum, it's nowhere near as sensitive. So only in 70% of those patients who are positive do you get a positive result. So there's quite a number of false negatives. So as a test, it's okay if it's meningitis, not as good if it's disseminated and you're testing serum. Also, be really wary of the idea of prozone. And everyone's going to go, what's prozone? Is that right? Please, you don't know, do you? Yeah, because when I was a boy being taught here, the term prozone was what everyone used, but now they talk about antigen excess. So imagine you've got a some latex particles with antibody to your antigen. What you want to have is antigen latex and the other side latex to cross-link. If you have so much excess antigen, here's your particle with antibodies all over it, you have so much antigen it swamps the whole thing and can't cross-link. All you end up with is a whole pile of particles of latex that are covered in antigen but no cross-linking and therefore no agglutination. I was taught that in immunology as the prozone effect. And every year I say prozone, everyone goes, never heard of it. So it just means an extreme excess of antigen which stops cross-linking. So sometimes you take some spinal fluid from somebody who's heavily infected, add your reagent, mix it, no agglutination. You might need to do a 1 in a million dilution to get agglutination. That's because the bug can be there in such enormous numbers, you can get a false negative because there's too many bugs there, or too much excess of the capsule. There are a couple of things that also can cause false positives. Don't overdo this, but just be careful. Trichosporin basilii, this is a yeast. It can also cause disease if you're immunocompromised, often from contamination of venous lines if you're on drug therapy. That will cross-react with the test. Agar will cross-react with the test. Now, how the hell can you get agar onto your slide for agglutination? It's like this. Here's your tube of CSF. You spun it down. You want to culture your CSF. Loop in the CSF. Agar plate, streak it out. Same loop back in there onto your slide for agglutination. Add the antigen, mix it. False positive. Now, you would never be that dumb, would you? You would either use a separate instrument or you'd flame your loop or do something, but just be wary of that. If you're positive for rheumatoid arthritis and you've got rheumatoid factor in your blood, you can give a false positive. Rheumatoid factor is it an anti-Ig or an anti-IgG? Rheumatoid factor itself is an autoantibody that you make as part of the disease that will actually bind other antibodies. So if the latex particles are covered in antibody, it will bind those and cross-react. So be careful about that. What's more important for us now is think about this. If you are HIV positive and we want to try and get to you before you become sick, the sooner we treat you, the better it is. If you're HIV positive and your CD4 count is below 100, so they monitor this CD4 count all the time if you're HIV positive, part of your monitoring your disease process, and we screen you with the antigen test, often you're positive 3 or 4 weeks before you become sick. So screening you means once you're positive, we treat you now before you're sick, and it requires less antifungal therapy to treat you before you're sick when the load of bug is low. It would be for you to get sick and then treat you. And you get much, much better success if we treat you before you're sick. Get in while the load is low. This is almost like a de facto kind of prophylaxis. I mean, you're already infected, the bugs will be there, but we're treating you before it gets established and you get too sick. And if we do that, then we have a much higher survival rate. So it's become part of the way you monitor and manage HIV patients, is to do this test as a routine if the count gets below 100. Back to that. It's kind of interesting. So we do see this in Australia as an infection. And if you look at the rate of disease in those who are non-Indigenous versus those that are Indigenous, and look at the distribution of cases between Varnea formans and Vargatii. So for Varnea formans, it's about twice as likely if you're indigenous than non-indigenous. Hard to know why this is. Probably it's more common here, indigenous than non-indigenous, purely because there's more HIV, there's more really poor health. I mean, these people are often not very well, alcohol abuse, poor living conditions, etc. But what staggers me is this enormous increase from Vargatii to indigenous. So it's much more common here if you're indigenous than not indigenous. I wonder why it's more common if you're indigenous to get infection with Vargatii. You think of a reason? The tree one. They hang out under trees. Seriously, they camp out under trees. They're much more commonly exposed. And you see the cases of the Northern Territory, it's more often seen in those that what we call campers. Yeah, it's all natural stuff. The ones that we see getting infection, these aren't the ones that are wandering around really happy and healthy. These are the ones that are down and out that live in the riverbank. But when you spend time in Alice Springs, for example, there's almost two lots of them. There's a whole lot of them that live in houses or on the missions and all okay, and you see them in town. And there's a whole bunch that live in the riverbank, and they all drink a lot and they don't eat very much, and they're really, really not very healthy. They're the ones that get disease, and the reason they get so much of this is their exposure from camping out. Now, that's only a hypothesis, it's not been proven, but when you read the articles published from the Northern Territory, where they do a lot of this work, that's what they're trying to say. I think it's all about their lifestyle and where they live in exposure. Now, Vancouver Island. This is something to me, so amazing. So these bugs have worldwide distribution. Why would there be this massive outbreak on Vancouver Island, of all places in the world? Why this one island that's suddenly become a focal point for infection? So if we think this is a big rate of disease, so 10 cases per 10 million, 10 million, this is almost 40 per million, so almost 4 times as much disease here in Vancouver Island than it is in parts of the Northern Territory. And it started Vancouver Island, British Columbia. So Canada, off on the coast. Vancouver, yep. So what we're seeing there is an outbreak that's been ongoing since 1999. So there's 1999, and it's going up and up and up. It's kind of leveled out now. This is only 2003. It's still going, even like today. And we're seeing cases in animals and humans. They found that if they do samples of trees, there are no gum trees here, but there are a range of other trees that are native to British Columbia and Vancouver Island that can also act as hosts. And they sample around the trees and get positives. So half of the ash trees were positive, about a quarter of the cherry trees, and about a third of the fir trees and oak trees in those islands were positive. They also did sampling of the air and found that the spores in the air were more common in summer than winter, linking to the flowering of the trees. And they were finding 30 CFUs per cubic metre of air in that environment. So air, 1 cubic metre, like about this much, there's 30 of those yeast in the air suspended. That's a lot for a bug like that. 10,000 CFUs per gram of soil under the trees. Now, why did it suddenly become so common all of a sudden? And they're kind of saying now that possibly climate change has raised the soil temperature enough to allow the bug to reproduce more quickly. So probably the bug's been there forever in that environment, some little niche that's been unique, but only in small numbers. When it becomes able to reproduce in the soil because of a higher temperature, it's now become so poorly controlled. And here's a map. So I've got terrible geography. So there's Canada, this is British Columbia, and off on one side is Vancouver Island. And on this graph, the red dots are human cases. Look at the number of cases in humans. And little blue dolphins, dolphin cases, animal cases. So we're seeing enormous amounts of disease over here and some disease over here. And when you map the wind direction, it goes from here to there. Cool, isn't it? And here's some, a graph shows you some sampling. So the open circles are tests that are not positive. So there's almost nothing over here. The ones that are permanently positive, so always positive, are the little yellow circles. Lots of strong positives over here, and only a few over here that are transient positive. So the bugs over here, it's been taken that by the air, by the wind currents. So here's a couple of examples of how you might see the bug presenting. This is one that's kind of nice to show you a bit like how the bug might present and how it's not always a disaster. So this person is reasonably old, 75, that's very old. Got a cough, non-smoker, HIV negative is really important. And diagnosed with X-ray as pneumonia. So do an X-ray, find a big patch, you've got pneumonia. What's this? Yeah, what kind of antibiotic is it? Erythromycin? What kind is it? Is it a penicillin? No. Is it a cephalosporin? No. Is it an aminoglycoside? Close, because they all end in mycin, but it's not. It's a macrolide. So it's in the same class as erythromycin and azithromycin. So if you've got a macrolide, what are you aiming at? What kind of bugs are you aiming at? Think about this patient. So this is a 75-year-old person who's not in hospital. What kind of bugs cause pneumonia in this kind of people? Strep pneumoniae, you're aiming really at strep pneumo. Macrolides are really good for gram positives, not so good for gram negatives. So it's really aiming at strep pneumo, possibly Staph aureus. No better after a week. Do a sputum, do a gram stain, see the yeasts. And you grow Gattii. This is the one you see more often if you're not immunocompromised. That's a reasonably high titer in the serum. So to be not overly sick, got a bit of a cough, it's not as though he's at death's door. Not spread, not in the CSF. Fluconazole is a reasonably good antifungal drug. Still positive though, so the bug is still going to be there. Took 12 months until he was screen negative, but survived. So, nice ending. Not so nice ending. This is one of the reasons why you've got to get in quickly. So, 25-day history of fever and headache. So the headache's already saying probably had some involvement in the central nervous system for at least a month-ish. So, you know, if you've got, you will never see someone going to a doctor and saying, I've had strep pneumo meningitis for a month. I'm feeling really sick now. With strep pneumo, it's in your brain today, and you're either dead tomorrow or you're not dead tomorrow, but you've got to be treated. You just don't have a month. This thing can stick around, it's chronic. But by then, unconscious, soon after admission, the white cell count's not overly high. I mean, 30 is not high for a white cell count for someone who's got a really bad infectious disease. And remembering that you get much more of a strong response in lymphocytes than you do in neutrophils. a count of 80 in spinal fluid is pretty high, so normal range in my mind is still per cubic millimetre is less than 10, so 80 is reasonably high. If that was bacterial, you might be looking in the thousands. So it's, again, not a massive response. Anything positive, that's quite a low T today. 1 in 16 is not as high as you would have thought it would be. And it's var grubii. This is the one you see more often if you're HIV positive. But he's not HIV positive. Here's the killer. Of all the drugs you can have that are systemic fungal disease, there are those that are really heavy hitters but toxic. And amphotericin B, like a sledgehammer, it's really, really aggressive, great drug, but not very well tolerated. It's quite toxic. And the literature says that you would treat this person with this disease with a mixture of amphotericin B and these two other antifungal drugs. And if you have amphotericin B with those other drugs, you get much better response. This person only got that one because this wasn't available in India. So often it's a matter of where you are and what's available. It can also dictate how well you respond. And he died. So to summarise infection with Cryptococcus, it's like this. It's very strongly linked to HIV and other kinds of diseases that make you immunocompromised, but it can occur in a sporadic way in people who are fairly immunocompetent. Diagnose it early. Microscopy from body fluids is not that good. You want to do a screen for antigen in body fluids, and that you get a much stronger positive much earlier in the disease. Culturing is pretty straightforward, grows really easily. Once you've grown the bug, identification is straightforward, either with MALDI-TOF or with microscopy showing the capsule. It's the only yeast, well, one of the only yeasts that's got a capsule, and it's the only one that's got a capsule and is also positive for fetal oxidase and goes brown in that media. Okay, let's have a 5-minute breather. So it's, by my clock, it's 10 to, stopped again. So 10 to 10, we'll start again at 10 o'clock. You wait. Last night, I saw that he started about 3 o'clock. I thought, wow, we're doing that tomorrow morning in the lecture. So I've added that slide onto today's stuff. But it is, it's pretty amazing to get that so quickly. So that's such a new bug. I only heard of that last year. In fact, last year, one of the students did a case study on Candida auris. And it sort of thought, oh, that's pretty interesting. One of those things where it sounds really awful, this horrible superbug, but really it's only a candida. It doesn't do much, but because it's so resistant, once it's got you, you're screwed. You can't get rid of it. Oh. Oh. Okay. Ready to go again? How you going, Jared? On this side, just do. All right. So, of the yeast, the most important yeast by a mile is Candida albicans. So the term candidiasis means infection with any kind of candida, and there's more than one candida species, but by an absolute country mile, Candida albicans is the most common, is the most nasty. You might just say it's only candida, but seriously, I do not want to get a serious candida infection because it's not a nice bug. So of the candidas, Candida albicans, and then there's a whole list of also-rans. Of the other species, Tropicalis is probably the next most important, and then the others really trail off in terms of how often you see them. But you'll see these other candidas in various kinds of patients who are immunocompromised. And the reason I've underlined Tropicalis and glabrata is these are both candidas that can be azole resistant. So the term azole refers to a whole class of drugs, just like we say penicillins, we say the azoles. And there's fluconazole and miconazole and ketoconazole. These are all just variations of the same basic drug. And these are the mainstay of therapy for candida infection. So if your candida is azole resistant, it means it's much harder to treat you. So unless I tell you otherwise, when I'm talking about candida, I'm talking really about Candida albicans in most situations. So Candida albicans is a commensal. Everybody in this room is carrying Candida albicans, completely everybody. Most of us carry it in our GI tract. It's very, very common in your bowel. And in fact, in a few weeks' time when we do parasitology, and I'm training you to find round things in poo that are parasites, the biggest hurdle is all you see are round yeasts. And you want to say, oh, that's a parasite. And I say, smack, no, that's a yeast, and you keep looking again. So they're always in your bowel. They're common. A smaller number of them you'll find in your mouth, and anywhere from 5–10% up to a third of women carry Candida albicans in the vagina, but in small numbers. So if we do a swab and culture, we can occasionally find small numbers of Candida in the absence of disease. You don't find many albicans on the skin, so it's really mucous membrane organism, gut, vagina, and the oral cavity. But there are other kinds of yeast you find on the skin, including the one that we've seen in a few people's toe cultures, which is the orange one called Rhodotorula. Almost always, infection with Candida albicans is endogenous. It means you've got infection with your own yeast. The exceptions are a couple. Sometimes men get Candida infection on their penis and they catch that from their partner. Men almost never give Candida back to the female because in men what you need is a penis and some Candida, and the bug being there is enough to cause infection. In a female you need a vagina and some Candida plus some underlying condition that allows the Candida to become dominant. Having the bug there is not enough. So if we just give you the bug, nothing happens. It's got to be an underlying condition as well. So really it's more common to see this in men as being exogenous. And we are seeing Candida now in a nosocomial setting, and even then, in hospitalised patients, most candidiasis, which disseminates and causes candidemia, like blood infection with Candida, it's still your own Candida that's come from your bowel through breaks in your bowel to cause candidiasis. Very occasionally it might be a common source outbreak via some kind of catheter tip or something else that's contaminated. So really, as a rule of thumb, it's endogenous for most people. A whole range of reasons why you might get Candida infection. Often they can be physiological. So things change in your body when you're pregnant. You have changes of hormones, and some of these hormones actually increase the expression of receptors that allow the Candida to attach. In infancy, you've got a very immature immune system, and Candida can take advantage of that. If you're diabetic, two things happen. You have more glucose in your body secretions, and Candida likes glucose. Also, if you're diabetic, you've got poor immune function, particularly for neutrophils, and Candida likes to be controlled by neutrophils. So if you've got poor neutrophil function, you're at risk. If there's Candida hanging around and there's a barrier break, it takes advantage of the barrier break. So we do see maceration really refers to a moist body surface that's been compromised by friction. So the most common sort of place to get maceration would be between your thighs, where you're a bit chubby and you rub and it gets moist and soft, and you might get Candida infection there. The term intertrigo refers to any kind of infection of the skin at opposing moist skin surfaces. So buttocks, thighs, under the breasts, under the armpits maybe. And paronychia is the infection along the side of your nail. It's really painful. That's often Candida. Two of the most common things that cause infection there, one of the reasons you get it is you might bite your fingernails, or you have your fingers being wet all the time from some occupation. It's either an anaerobe or a Candida. It gets all red and pussy. Hospitalised patients, IV catheters are a major source of Candida. Long-term catheter in place for blood monitoring or drug delivery, you might get candidiasis. But unseen holes in your gut wall. So when you're really, really sick, you're talking about someone who's bedridden, they're on long-term drug therapy, could have cancer or something. There are holes in your gut, you can't even see them. If your gut's full of Candida, they leak in and you can get candidiasis, as well as gram-negative sepsis as well. There are a whole range of specific immune disorders that make you at risk of candidiasis. So diabetes, HIV, any kind of cancer, especially blood cancers. So on the haematology ward at hospitals, there's a big risk of candidiasis. And then the last bit, which we're all probably more familiar with, is if you have widespread antibiotic use, there's a risk because you disturb the normal flora balance. Antibiotics kill the bacteria, you remove that competition, you remove the things that they produce that are anti-Candida, and in comes Candida. This is the most common thing that precipitates vaginal thrush in young women, would be the use of antibiotic therapy. One of the most striking things about Candida is you often see Candida in urine specimens and you think it's a coagulation negative staph because the colonies are white. So whenever you see, and this is going to happen eventually, if you see somebody reporting from a urine a coagulation negative staph that's totally drug resistant, it's a Candida. Because there is no such thing as a coagulation negative staph that can resist every antibiotic. But if it's a Candida, all those drugs are antibacterial, it'll be fully resistant. And you often see people make that mistake. Because really, once you leave here, the notion that you will do a gram stain on a colony is just like bizarre. You'd never do one. The number of times a microbiologist will do a gram stain on a colony in a real working laboratory, once a week. It's all like, that's what that is, and you do it based on visual stuff. There's not much, there's this gram stain going on, which is why I try and win you off doing gram stains all the time in our practice, because no one does them. Here's a range of diseases that we do see. So it will range from disease that's cutaneous, that is only on the surface, to those that are mucocutaneous, which means it's on the surface plus mucous membranes, to disseminated. The top one, not a big deal. You do see skin disease. You do see it in babies who've got, had nappy rash. Don't you hate the word diaper? Oh, do you? It's a nappy, not a diaper. I've changed hundreds of nappies in my life, never changed a diaper. Bloody diaper. You do see Candida causing disease in nails, especially if the person has any kind of underlying immune disorder. And in the nail, it'll just look like a dermatophyte. So the nail itself appears to be dystrophic, it'll be lumpy, it might be a bit discoloured. And you do microscopy, you might see a little bit of pseudohyphae, but you won't see long hyphal strands. So the microscopy will be different, and the culture is different, but clinically it's the same. You would think that might be a dermatophyte. It wouldn't matter because the drug therapy for dermatophytes would also cover Candida albicans. Once it goes from being cutaneous to being mucocutaneous, it's much harder to get rid of. So we do see oral thrush, especially in young kids if they've had antibiotic therapy. If you've got underlying immune disorders, it can become chronic, and you see children who've always got a white plaquey sort of thing in here, each side of the lips, and that's just Candida that's going in their mouth in sort of low numbers, and it'll be on their skin, might be in their fingers here, it might be in their gut, but it can become really chronic, and you can't do much about that other than restore the underlying immune disorder. So in men who get Candida, it's really going to be on the head of the penis, on the foreskin, and all that happens then, it's just like a skin disease. The skin happens to burn your penis, but it's not very nice, a bit painful. But I can't talk with authority about vaginal thrush, wouldn't even attempt to. But you do see that it occurs most often when there's excess of estrogen. So when you're pregnant, maybe when you're on hormone replacement therapy. And you can treat this if it's only an acute disease that is a one-off or two-off episode with topical therapy. So either creams or pessaries, little tablets that you insert that explode gently to relieve. I hate to say that. But here's the big clincher. If you're unlucky and you get repeat episodes, like you get Candida this month and you treat it, and there's some more next month, and there's some more next month, then it might be important to go on prophylactic therapy. That is, take the azo drug for a long period, like a couple of years of having low-dose azo therapy. Now, the risk of having low-dose azo therapy is you will select out either mutants of Candida albicans that are azole resistant, or you select out from your bowel strains of other Candidas that are naturally azole resistant, like Tropicalis. And then once you've got that kind of chronic condition, it becomes harder and harder to manage all the time. And then of all the ones you don't want to get, Candidemia, this is so bad for a couple of reasons. The first reason is to get Candidemia means your underlying health is already bad. Healthy people don't get Candidemia. You've got to be sick. Once you have it, it's really hard to diagnose. There's no great test that goes, yep, that's Candidemia. So a blood culture can find E. coli like that, but finding Candida doesn't grow well in broth. So blood cultures are not great for finding Candidemia. And then once we find it, removing the Candida becomes more and more difficult because the drugs are not great. So you don't want to get Candidemia, obviously. Sorry about this. So, white patchy discharge of a Candida. And I'm always amazed by this. This is meant to be short and curly. They must have used a hair straightener. It's too straight, isn't it? Shouldn't be like that at all. It's horrible. So, that's all Candida. You can imagine how painful that would be. That's on the penis, so that's on the head and back there, very painful. So You might just go, oh, it's only Candida, but seriously, Candida albicans is a nasty bit of work, especially if you're immunocompromised. So that's all up the thigh on the scrote, be very painful, very painful. Now we're seeing someone who's immunocompromised, and that is on the roof of their mouth. If you took a swab now and you just did that, this plaque would come off. If you took that off and did a squash prep, all this white would be a mass of Candida cells and pseudohyphae. You can actually buy Candida toothpaste. I would love a jar of this just as a souvenir, because Candida means white. So in some languages, Candida means good toothpaste for white teeth. But I just can't imagine it using that in my mouth, to be honest. That's Candida. That's paronychia, so infection along the side of the fingernails. That's chronic candidiasis. So that's clubbed of the fingers. This person's probably immunocompromised. That's all Candida albicans. As a bug, it's so flexible. As a baby, Candida albicans. Next one's not very nice. That's a baby who's born with systemic candidiasis. The baby was immunocompromised, but again, look at the bug, how versatile it is. So even though we think, oh, it's only a bit of Candida, I can't stress enough that it's a nasty bug. Chronic disease in the finger webbing, so this is always moist in there, that's Candida. Here's a person with chronic disease on their tongue, side of their mouth, and their fingernails. This is a person who's not immunocompromised. That's their toe spaces. And you can imagine if you were to do this, it would peel off and it would smell. I love this. Now, this is Candida. This is the same person after two things happened. First thing happened is they had some azole therapy. But the most important thing was, at this point here, they were heavily immunocompromised. They were on tumour suppressive therapy. And over here, they removed the therapy. So between there and there, the real thing is immune response. Immunocompromised, Candida is nasty. If you're healthy, Candida's nothing. Also Candida. So again, person who's immunocompromised, Candida on the scalp. HIV positive patient, Candida is disseminated and it's come from the blood to the scalp. This is looking in someone's eye. This is Candida retinitis. So it's come from the blood into the back of the eye. Are you convinced already about Candida? It's not nice. So what do we do in the laboratory? What's our role for candidiasis? If the disease is simple cutaneous disease or even mucocutaneous disease, it's fairly straightforward. Direct microscopy is pretty rewarding. It's not hard to find, and when you see them, they're pretty distinctive. Candida is a typical oval yeast that buds. It's an oval shape, not round, it's oval, and the buds are little babies that bud off. When they bud, as the buds grow, sometimes they grow and don't snap off, and we call that pseudohyphae. Pseudohyphae means there'll be a point of constriction between, so if you've got this, that's real hyphae. What's the light's going on? Oh, there it is. But this with constriction, that's pseudohyphae. And you see pseudohyphae, not true hyphae, with Candida. You culture it, it grows on lots of different media. I mean, Candida will grow on Macconchia agar, grows on blood comfortably, We use SABS agar because SABS is slightly selective. The idea that SABS won't grow bacteria is rubbish. It just grows them poorly. So the extra sugar in a SABS agar supports Candida in preference to bacteria, but bacteria still grow. You get Staph aureus Nicola on SABS agar comfortably. But SABS works really, really well. Now, gonorrhea media. So this is like how you screen now in vagina swabs. Gonorrhea media contains a whole range of inhibitory compounds to make the plate selective, including an anti-yeast compound called nystatin. And it seems bizarre, but you can actually use gonorrhea media to be selective for yeasts in a strange way. Like this. The drugs in gonorrhea media that inhibit bacteria are very good. So you won't get growth of enterococci and coliforms and pseudomonases. They won't grow at all. The inhibition of the yeast only lasts for about 24 hours. So at 48 hours, the yeasts will grow on the gonorrhea media. But the growth is changed because of the stress that they're under from the nystatin. And they grow as colonies with feet. So rather than being a lovely round colony in profile, looking down on top of the plate, they're like this. They have all these little projections that run up into the agar, like this. When you see those little colonies with feet, it's almost a way to screen your plate for Candida. And some labs have moved away from using SABS agar and use the gonorrhea plate to save money. Why don't you? You should. Yeah. It would make sense to have SABS agar with chlorphenicol. Hard to know why you don't. In most labs, you can have SABS agar with chlorphenicol, and that's all you ever do, because there's never a need to grow anything other than fungi on the SABS plate. So why wouldn't you? Yeah. Now, because we're getting so good with our chromogenic media, the preferred media now for culturing vagina swabs is a chrome agar. So a chrome agar for urines we've got developed where you just put urine on the plate, and if it's blue, it's E. coli. Well, there's now a plate like this for Candida. And you do your vaginal swab on this plate, and if the colony is there, I think they're blue, they're Candida albicans. So the preferred screen now is to do away with all the other rubbish and just go straight to a chrome agar. They are expensive, so per plate it might be, I'm just guessing, $1 per plate maybe. But because all you've got to do is look at the plate for a colour and you give it a name straight away, they're good value because it's quick, as opposed to the other ways of doing it. So these are quite good. Now, these plates are a little bit selective, and they will also identify other kinds of Candida. So you'll get an ID for Candida albicans, and I think for tropicalis and a couple of other Candidas. So there's more than one colour on the chrome agar plate, so they're very good for the common Candidas from vagina swabs. If you don't use that media, and I can't imagine why you wouldn't do that, but in some labs you won't be using this media, it's too expensive. If you're doing plain SABS agar or gonorrhea media, then what you get is growth, and you say, I have grown a Candida. Then the question is my Candida Candida albicans? And the most common test historically to prove your Candida was Candida albicans is the germ tube test. These are really easy to do. The germ tube test says take some serum or some plasma or some special broth, but serum is better. Take some serum, put a bit of your colony in the serum, incubate for 3 hours. Take some stuff out of your tube onto a slide, microscopy, have a look. And a germ tube is when the yeast makes a projection called a germ tube, like that. Now, if you see this, not a germ tube, this is a blastoconidium. The germ tube is not allowed to constrict at the point it comes off from the candida. If you wait for more than 3 hours, you get false positives, but at 3 hours, it's a reasonably good test. Except now we do know that there are a small proportion of Candida albicans that are negative by the test, and there are other candidas that are uncommon but also positive by the test. So as a test, it's okay but not perfect. I think we've got to realise that most times it's all right to be okay. You've got to be perfect. Other tests that we can use, older test was called an oxanogram. Now, an oxanogram is a horrible test to do. An oxanogram is a bit like an API. And what you do is you have a whole range of carbohydrates as a substrate, and you have this carbohydrate with a bit of broth, and you put the candida in there. If the candida can assimilate the carbohydrate and grow, it's called a positive. And the way you look for a positive, it's got to go cloudy, and a negative is not cloudy. What you call cloudy and not cloudy is really poorly defined, and the test used to have a little black, little cup that was colourless, and at the bottom of the well were some black lines, and it said, if these black lines are easy to see, it's negative. If they're hard to see, it's positive. So when you used to go and mark them out, like plus, minus, you'd always go plus, maybe minus, possibly plus, and in the end it was a nightmare. So I find these tests really hard to do properly. And the good thing is, MicL4 now has removed the need to do these tests. So if you haven't got MicL4, you do a germ tube. If you've got MicL4, you do a MicL4. What's much more important though is when do you bother? So you've grown a yeast. Not all yeasts are Candida, but some of them are. And not all Candidas are Candida albicans. So when do you identify to the nth degree, and when do you start making guesses like this? If you've grown your yeast from an otherwise sterile site, you must do a full identification. You just have to. Go all the way. That might be MicL4 or something else. If it's a cutaneous disease, or if it's vaginal infection, it's probably okay just to use chrome agar or a germ tube and use those to call it Candida albicans. And if these are negative, then you might just call it Candida species, brackets, not albicans. Sometimes, if it's not shown to be Candida albicans, and you're not comfortable with just saying, oh, it's Candida, but it's not albicans, you might then go one step more and do full identification. But only with a good reason to. If you had, say, a first ever case of vaginal thrush and someone said, you got Candida but it's not albicans, you wouldn't care whether it was tropicalis or prapsilosis or glabrata, couldn't care less, doesn't matter. But if you had recurrent thrush, you'd care, because then if it was tropicalis, it might be that it won't respond to azole therapy. So you might want to know more about it then. Otherwise you wouldn't. So the length you go to depends a lot on what's the clinical significance of the isolate. Simple one-off episodes of cutaneous disease, it's reasonable to do not much. A bit like E. coli with urines. Most times it's okay just to go to E. coli based on very limited evidence. Sterile site though, you must do full identification, have to. And sterile site, probably you couldn't use chrome agar to be comfortable. Chrome agar is good, but there are a couple of other candidas that are not albicans that are important that look like albicans on chrome agar. And we'll talk about those in a minute. On microscopy, you'll see a mixture of bits of pseudohyphae. Now, on, say, a prep from some skin or something, you can't distinguish hyphae from pseudohyphae. Don't even pretend you can do that, you can't. But what you're seeing there are lots of yeast cells, and these couldn't be Canidia from a dermatophyte, because they don't form spores in vivo. So really, once you saw this, it's got to be a candida. Blood cultures. What if you've got disease that's disseminated? You've got candidemia. Candida is aerobic, and blood cultures are really only good at growing bugs that are facultative. Now, if it's a Pseudomonas and it's aerobic, it grows quite well. Some bugs grow well in broth. Yeasts and moulds do not grow well in broth. So a candida in a broth grows on top of the broth, like a little pellicle, little top lid almost to your broth. So we find that if you do blood cultures, it takes a lot longer to go positive than it would have been if they were bacteria. So it might be strongly positive, but take 3 or 4 days. And if you've got small numbers of yeasts in the blood specimen, it may remain negative and not go positive, especially if you're using a blood culture system with detection that's automated. They require things like gas production and change of pH and change of conductivity. So unless you're doing blind subculturing where you'll find small numbers, often they stay negative. So if you're a big laboratory that services people who are immunocompromised, say big haematology wards in the hospital, you can buy a system that's dedicated to growing fungi from blood. And the way it works, the machine's called an isolator. And what they do is you take the blood, but rather than put the blood into some broth, you take the blood and you lyse the blood, and you spin that lysed blood down, and you culture the pellet of the spun down lysed blood. That works because fungi grow better on a plate, on a solid surface than they do in the broth, and lots of these fungi and the candidas can be intracellular. When you lyse the blood cells, you're removing those bits of fungal material from inside the cells to make them then become available to culture. So without specialised systems, blood culturing is bad. What we need, really, is a different way of finding candida in the blood. And you can go and do a search in literature, which I do every couple of years to make sure I'm not missing something. And they are looking really hard for other things to find candida in the blood other than culturing it. What they want to find is some metabolite of candida, and if you find this metabolite in your blood, it goes, you've got candida in the blood. They're trying to find some unique thing, some antigen maybe, or some metabolite which is a marker of candidiasis. And right now, what they found are either not very sensitive or not very specific, and they're still looking. So as of last year, I had a good look again, there wasn't a good test for candidiasis beyond just doing culturing. And they're germ tubes. Nice little germ tubes. We'll see these in the practice for a bit of fun because they're nice to look at. So these yeasts look more round than normal for Candida. Candida is normally much more oval shaped than that. But there's a germ tube positive. Forget that, I'll test out of the syllabus now. Should remove the slide until next year. And there's your chrome agar. You'll see that albicans is a reasonably straightforward colour. This greenish blue, tropicalis, different colour blue. So these two are the two most common candidates from vaginal swabs. This plate really is about doing vaginal screens. This is not a plate you would use if you've already grown your candida from somewhere else. It's not a tool for identification really, it's more of a tool for screening. And in that unique situation, we're happy to use this degree of evidence to call it albicans tropicalis. You wouldn't use that on a blood culture isolate. You just wouldn't. It's just not reliable enough. Right now, every routine lab is really good at testing bacteria for drug sensitivity, but almost no routine labs test fungi. One of the reasons is that most times, if you've got a simple candida from a simple single disease, the risk of it being resistant is very low. One of the reasons why candida tends not to be so resistant and it be so widespread is bacteria can exchange genes. So with bacteria, they either mutate themselves or they acquire genes from somewhere else. With candida, it's always their own genes that are mutated. There is no transferable difference in candida albicans or any of the candidas. They don't do it that way. There's no exchange and recombination. The only time you see these strains being resistant is if you've had somebody on long-term therapy already that selects out mutants. It's not done that widely. You do want to test candidas, you can't do disc diffusion. There's no calibration for disc diffusion for candida. You might be able to do a screen where you put a disc on and look for a certain zone, and if it's all that will do is say this is probably one that's azole resistant. You've got to do MIC testing in dilution series. So big labs will do this, but not small labs. So as far as I'm aware in Tasmania, no one does this testing here. Was it? Spelling error, I hate that. Where? Oh, it's not my slide. See, I wouldn't have written that. I'd have picked that up for sure. It was my slide. I borrowed that from somebody else. That's from David Ellis. I must tell him. Now, there are two new candidas on the block that you have to be aware of. So mark these in your little mental head and go, yep, got to remember Dubliniensis. So Candida Dubliniensis is quite a new one. It's been around for maybe 10 years, maybe 5, more commonly. It looks just like Candida albicans on most common media. So even on the germ tube it's positive, on the chrome agar it's a similar colour to Candida albicans. You would call this Candida albicans every day of the week without some suspicion. But it's not Candida albicans, it's a different kind of Candida that we see really as an important pathogen now in HIV patients. The risk of you finding one of these and not being aware is pretty minimal as long as you follow this kind of scheme. So if they were HIV positive and had vaginal thrush or had thrush on their penis, it wouldn't matter whether you called it albicans or not. No one really cares. We're talking about this being systemic candidiasis. And if the candida is systemic, then the rule of thumb says sterile site, do a full ID. And multi-top is good at differentiating them. What if you're in a small lab that hasn't got a multi-top? How do you then do this? Well, there are other tests to do. So one is you can do subculturing at 42 degrees Celsius. Candida albicans grows well at 42, and this grows poorly or not at all. And not a great test. This whole thing poorly or not at all I can handle with, but poorly, to me, either growing or not growing. this poorly grows. What do you call poorly growth? You can take a caffeic acid agar and subculture this on caffeic acid agar. Candida albicans grows normally on this agar, doesn't turn brown, but just grows normally as a normal colony. And Dubliniensis grows as a rough colony with spiky edges. I think the sooner we all have Molitod, the better. Oh, spelling error. Molitod. Where's the D on the keyboard? Is it the same? Is it close? Where's the F and the D? Oh, next door to each other. There you go. That's because last night doing this on the couch at about 11:30, I wasn't being very careful. So is that on your slide? That line there? See, I checked last night to make sure it was up to date. I hope you're grateful how much work I put into you guys to make sure you're all up to date. Hey? Oh, you can write one line, can't you? Yeah, when you write this down, write down Molitod, not Molitod. Now, Shaylee, here's the one that's interesting. So yesterday on the ABC News website, they made a mention of this. So the new Candida called Candida auris. The reason this has become so important is it started off as being simply a bug that caused an ear infection in Japan almost 10 years ago. And what happened is it spread from there to now being seen as a case that disseminates. So we've seen these candidas in patients who are immunocompromised in their bloodstream. But it will resist all the azole drugs. You can't use fluconazole, you might try ketoconazole or some other conazole, and they all won't work. If they won't work, you might try the old fallback amphotericin B, and that won't work. Then you try the newer drugs called caspofungin, and that won't work. All of a sudden, all the drugs that we use to treat systemic candidiasis don't work. So Candida auris is like a super version of Candida albicans. It causes the same kind of diseases, it's just that it's inherently multi-drug resistant. That's all there is about this bug that makes it super. No worse disease, it's not more aggressive, it's not anything fancy, it's just hard to treat. Because we are not good at doing tests for drug sensitivity, the best way is to find out if your Candida is albicans or auris. If it's auris, you know this already applies and you'll be much more careful with how you treat them. So, as of last year, when I put this slide together, it's been found here and here and here and here, etc., but not here. As of last night, there's now a red dot there in Victoria because yesterday on the news, they said they found one in Victoria. The person had travelled overseas, acquired the bug and come back. The good thing is the person is not infected, they're only colonised. It does tell you though that people are worried about Candida enough that they're now screening people to see who's colonised. Now I must try and find out how they do the screening. If you were to screen these people for Candida, what would you do? So where would you become colonised? Be in the bowel, wouldn't it? So you screen the bowel for Candida. Well, everyone's positive. So what's the point? They must screen with a plate that will find Candida and also ones that are resistant. So maybe a plate like a chrome agar plate that's got, say, amphotericin B or fluconazole, and it's screening to see if you, not only are you carrying yeast, but you're carrying yeast that are drug resistant. I'm going to find out how they do it, because I'm just curious. It's a bit like doing VRE screening. If you screen someone's bowel for enterococci, what's the point? Because everyone's positive. You want to screen for ones that are vancomycin resistant. But the same here with the yeast. And you screen them. When do you screen people for bugs that are drug resistant? I mean, would there be any point doing a screen on me now to see if I've got VRE or MRSA? No point at all. You screen them when they're at risk of infection next week or the week after. So when they're compromised, maybe they're having surgery, maybe they've got massive burns, things that put them at risk. And if you know what they've got now that's colonising them, they're the bugs that might infect them next week, and you want to get ahead of the game so you've got a better plan for therapy next week. Okay, what's coming up for us? So, have a break, we'll come back and I'll go through the test with you, which I've marked. Thank you. And then we'll get a plan ahead. Coming up is Miss test, and coming up is the prac exam for mycology. With the mycology prac exam, it won't be hard because mycology is too hard to make it hard. Oh, seriously, if I gave you a Fonseca and said, what is that? I couldn't get it right either. So what the heck can I ask you to that? But I would say, can you recognise penicillin? And if you can't do that, go home. Can you recognise cladosporium? Simple things that are really quite straightforward. And not only that, I'm more inclined to want to know you can recognise and give a name to the right kinds of observations. If you said this, if I gave you a cladosporium and you went like this, round macroconidia, chlamydia spores, therefore cladosporium, right answer, wrong reason, 0. If you said there are branching chains of candida and rhamnosus and the ends have little scars on them and they're shield shape, not sure what it is, that's worth 8 out of 10 because you saw all the right stuff, you just couldn't remember the name. It's about seeing the right stuff. And quite probably, I'll give you one fungus in the exam you haven't seen before. Purely because I want you to shake your head, Aruba. Look at this, she's going like this. Because I want you to be able to see and say, yep, I saw this thing here, and I would describe this thing I'm seeing as one of these. So you might say, I'm seeing lots of piriform microconidia, and the ends are truncate. Can you observe and describe the observations properly is more important to me than can you just remember a name like that. In the end, you've seen some of these fungi so many times, you'll just go, oh, I've seen that one before. That was one of those. And you just write the name by association, not by being anything that's clever or any skill acquisition. So there'll be 3 or 4 fungi to identify. And honestly, unless you have been asleep in the pracs, I wouldn't see this as being a major hurdle. But if you think you can just go like this, oh yeah, seen that thing, brown thing before, that's one of those, it won't be enough. You've got to make sure you've seen the right stuff. Okay, so I'll come back here, say, at 11 o'clock and we'll do the test. Go through it. So question 1, talking about hyphomorphology and what's important. So really the key was, could you recognise that it's important to separate them into, is the hyphae dermatiosus or hyaline? That's worth 1.5 marks, roughly. If you only got that, I just only gave you 1, I gave you 2, didn't give you 1.5. And then, is the hyphae septate or non-septate? The 2 key things are hyaline dermatiosus, septate, non-septate. You just get those 2 things by themselves, that's 3 out of 4. The extra bit would be, can you think a bit naturally? And sometimes things like, does the hyphae break up into arthrospores? Now, even though this is a spore thing, it is really part of the hyphae, so that's probably worth an extra mark if you could see that. Maybe you might talk about occasionally having spiral hyphae is reasonable. You talked about the ones that are non-septate being really broad hyphae, that's also reasonable. So the first 2 points are worth 3 out of 4, and the extra 1 to get 4 out of 4, that's something a bit different that's relevant. You talked ad nauseum about all these conidia, it's just not relevant. Conidia aren't part of the hyphae. We're talking about hyphal morphology, not about other ways to identify bacteria, the fungi. Now, question 5, talking about what we're saying here is different patients And I made a point here of saying they correctly identify the bug as T. meningophytes. Because I've said that they correctly identify the bug as T. meningophytes, if you started wasting your time talking about maybe they were wrong, it was T. rubrum, you're kind of a bit pointless really, because I've made a real effort to make sure you know it's the right identification. So don't give me any argument about T. rubrum and they make a mistake. The first thing is, I would say, is there are different varieties of T. meningophytes. So inherently, one species has more than one variety, and they will have different morphology. On top of that, even the same variety, there is so much variation in how they present within microscopy that the very same variety, but different isolate, would be different. So we're saying these two people could have different organisms. It may be a whole different variety. It might be the same variety but a different isolate. And then, how about the part of the colony? We know that the part of the colony you sample for microscopy is really important. Maybe this person took the middle of the colony, which is really old, and this person took the edge of the colony, which is a lot younger. Colony age is important in your microscopy. How about the type of media? What if one person took their microscopy off cornmeal agar, you did yours off Sab's agar, or they did soil and you did lactic meal? The agar is so critical. You must make sure it's the same agar to compare between different people. And what if they did a different preparation than you did? What if you did a tape prep, they did a siege prep? You'll see different stuff because one will retain the architecture and one won't. The whole point of the question was to understand that variation is really normal in mycology. Things always being the same is abnormal. You've got to get used to why things will change. If you know why things will change, then you'll make sure that you employ systems to make sure you control as much as you can to minimise the variation that you see that you can't control. So I always use the same media. I always use the same part of the colony. I always use the same age of the colony. And that way, what I'm seeing, I can relate back to last time I saw it and the time before. If you just do any old prep of any old media from any old age colony, you won't build up a memory of what you to look for and what means what in your own head. So they're roughly worth about 1 mark each for those kinds of points, roughly. Question 3 wasn't answered that well, and I hope it's not because the question's too vague. I tried really hard to make this question seem obvious, but maybe I didn't do a very good job of doing that. The zygomycetes, like micL4 and rhizopus, are put together because they are sexually the same. They make zygospores. Now, we don't see zygospores. In the lab, we see sporangiospores. But they're put together not because they make sporangiospores, because sexually they make the same kind of spores. So they're in the same group because they reproduce the same sexually. Almost all forms of classification are linked back to how animals reproduce sexually, including fungi. So zygomycetes are there because they make zygospores. They're in the same family for that reason. So the first point I'd say is dermatophytes have no sexual state. So you could not classify them by that system because it doesn't exist. So 1 mark for saying something like that. They don't have a sexual state. They're often called the fungi imperfecti. There's no sexual state. They're always asexual. So if they're only reproducing asexually, what do you do that links them together? Well, the first link with the dermatophytes is nothing at all to do with microscopy. It's to do with the clinical disease they cause. So they all cause cutaneous disease that involve them metabolizing keratin. Make a point of saying they're not sexual, and then talk about the clinical similarity in what the diseases they cause. And then you might say something like, the other similarities are that they are all hyaline molds, and they are all cyclohexamide resistant, for example. But really, they're classified together based much more on their disease causation than anything else. But there are other things that, there are other fungi that cause tinea-like diseases that aren't dermatophytes. So there's more than just that one property. They do also form fairly similar conidia in the sense that they often form more microspores than macrospores, depending on the genus. The real bit there was to make sure you get these things have no sexual state. Yeah, so they're in that group. So why did somebody decide that trichophyton and epidermophyton would be in that group? Why didn't someone decide to put epidermophyton in this different group? They're in that group. They've given that family name for a reason, because these bugs have similar properties. And the most common similarity is they cause the same disease. Now, we couldn't use that system in bacteriology because then you'd have all the bugs that cause meningitis would be in this special family to get a name. Well, that would be strep pneumo and Neisseria, and it would be so weird. There is no simpler way with the dermatophytes because taxonomically, there's no great connection to them. There is no great genomic link between epidermophyton and trichophyton, other than they cause the same kind of disease. You think about microsporum and trichophyton, they're so different in their morphology that genomically, if you did a genomic tree, I wouldn't mind seeing if there's one being done by somebody else, I reckon they'd be quite distant. So they're put in that family for that reason. That's the primary link. Does that make sense? Yep. Yeah, it's a family that's been designed for them. They've made the name up for them. So question 6 was really about, it's more about, can you explain why they're in their genera, spend less time explaining how to speciate them. A lot of you spent time talking about how to speciate them once they're in that genera. You didn't lose marks for that, but there's no real bonus marks in it either. So talking about, it's M. canis if it's got this, but M. if it's got this, that wasn't the question. It's about why they're in the genera. So all I would say is that, in general, to put them into that genera, so trichophytons tend to produce abundant microconidia and much less macroconidia. And that puts them in the family Trichophyton, roughly speaking. I might ask you general questions because you can't be too specific with that kind of question. They're in the family, or in the genus Epidermophyton, if they don't make microconidia, and they do make club-shaped macroconidia, and they do make chlamydoconidia. And they're in the family Microsporum if they make echianulate macroconidia, normally abundantly, not always, but normally abundantly, and they make fairly nondescript piriform microconidia. They're the things that put them into various genera. So if someone said to you, here's a microscopy, here's a slide, what genus is this fungus in? They're the things you would say in your mind to put it into different boxes, and then you do other tests to speciate them. So what puts them in those genera is the first part of the question. And I gave you 2 marks for each genus, basically. Now the drawing. These were hard to mark because I didn't want to penalise you because you were shit at drawing. But it would kind of make sense if you were doing a drawing to identify key features, it would help to label them. Next year, I will specifically say, please label your diagrams. Otherwise, you might draw a phyllide and not label it. How do I know that what you've drawn is a phyllide, you won't call in conidia, and vice versa? And some students who did label them drew conidia and called them phyllides. So sometimes you actually might lose more by being more efficient in doing labelling if your labelling is bad. There's a fine line between me being picky about your drawing and me letting you get away with too much. So sometimes if you drew a firelight that looked remotely like one, I gave you a mark. If you just drew a blob and called it a firelight that didn't resemble a firelight, there's no mark in that. If you just draw me a square thing and say that's a firelight, doesn't mean you're right, does it? Just means you call it a firelight. And I know how students' minds work. If you're confused, you make your drawing so small I can't distinguish what you're saying, and you hope that I'm just going to give you marks. So it says here, hyphae with septate, no mark in that. If you can't draw septate, it shouldn't be here. So we've got some hyphae. And it says the cnidiophores are lateral. So they're not being formed at the end of the hyphal strand, they're being formed laterally. This is lateral. And they're branching. So there's a branch. This is a branching cnidiophore being formed laterally. And it gives rise to single phyllodes at their tip. So the cnidiophores give rise to single phyllodes. So a phylloid is normally flask-shaped. It's a whole reason they call them phyllodes. And somewhere, in some Greek language somewhere, I'm sure that phylloid or fire something means vase or something, I don't know. Maybe in the Greek language they called phyllodes with 10-pin bowling pins, I don't know. There must be a word there for a reason. And it gives rise to single chains of microconidia that are piriform in shape. Piriform means pear-shaped. They're not round. They're more like this kind of shape. And so if I was doing this, I'd be saying branching cnidiophore phylloid, and then I'll just pull one of these off by itself and say piriform, not round. And then I'd know you knew what you were talking about. And you're going to form some arthroconidia. So I gave marks, basically, is did you make your cnidiophore be lateral? Did you show some branching? And did you draw a phylloid that was the right shape? And did you show that these were piriform and arthrospores? There were 5 marks for that one. Now I'm sure we can sit down together and argue to the cows come home about how nice or not nice it was with it, how much you're drawing. In the end, I've just got to make a judgment and just make an assessment. I'm sure that if I remarked every single question, I might go up or down by 1 mark. Get over it. There's no point arguing over that 1 mark because we can sit there forever and argue. If I've been really, really unfair, by all means, come and see me. I'm more than reasonable to make an error and admit I've made an error. But most times they were hard to mark because the drawings were a bit vague sometimes. I'm not a great drawer, but just try and make them clear and don't make them too small. Now it says septate hyphae, same again, no marks of being septate, that's a big deal. And it says lateral unbranching cnidiophores. Lateral, not branching. And it gives rise to clusters of phyllodes, both terminally and laterally. So on this cnidiophore, there'll be a cluster terminally and some clusters laterally. And the phyllodes are elongated with a slender tip. So it means that this time you draw the phyllodes should be different than the previous time you drew them. So I would draw these as elongated with a slender tip. And they're in little clusters. And I draw the same there, little clusters of them. Like that. Conidia are in clusters, not in chains, clusters. And they're oval shaped. And when they come off, they truncate. So I go like this. One's come off, I'll do it a bit larger. Truncate the bottom. Like you saw yesterday in which organism? Episporium. And there are occasional intercalary chlamidospores. That's it. So there have been a mark for this being lateral and not branching. There have been a mark for these being clusters terminally and laterally. And there have been a mark for this and a mark for this being truncate. Question 4. I even put it in bold and underlined it. Positive direct microscopy. This means it's a specimen, not a culture. If you wrote reams about eflacosin not doing microspores but having club-shaped macrospores, it's not relevant. That's only what you see from microscopy from a culture. All dermatophytes look the same on direct microscopy, and all you see will be bumpy hyphae. Septate, if you want to say that, it doesn't really matter because you won't see septate anyway on microscopy from specimen. So you see bumpy hyphae that occasionally breaks off into arthroconidia. 2 marks for that. And Taenia versicolor is the Malassezia furfur yeast in those lesions on the skin, and you see short angular hyphae and round yeasts. If you write down spaghetti meatballs, that's fine by me. Everyone uses that terminology. I'm not sure it really means much to us anymore. When was the last time anybody here has spaghetti meatballs for tea? Not in my memory. It's one of those things that just doesn't happen anymore, does it really? Although last night I had 3 nice meatballs on my way home from work. Mum's house, I called in to say hello and she had some meatballs there. I had 3 of those. For 3. Oh no, this morning I weighed myself up to 77 today. I am not happy about that. Even more now I've had that bloody hedgehog. It's a conspiracy. So, what's that? Yeah, I know. Okay, question 7 about microscopy, about the colony morphology. So it's like this. You want to look at the colour of the colony, got to be top and reverse. 1 mark for saying colour, top and reverse. Not 2 marks for that, just 1 for top and reverse. Texture. Is it downy or cottony? Is it granular, etc.? Profile or shape. In other words, 3-dimensional look. Is it flat? Is it lumped and rounded? Is it rugose? All that sort of stuff. So 1 mark for each of those. Extra mark could have been gained in lots of different ways. Super important is the media. As a rule of thumb, we describe colony morphology by default on SABS agar. And the bugs look different on different agar. If you try and describe them on soil agar, they're all flat and unhappy, look the same. So the media you use is really important. If you're comparing what you see on the phone to a colleague somewhere else and you say, oh, I saw this, oh, but I saw this, different media. Tell them what the media is. And then a lot of you wrote down size. No marks for size. Because size is relative to how old the colony is. If you say, oh, my colony is really small, that might be because it's a slow grower or because you're in a hurry. So it's the age is more important, not the size. How old is the colony? And a great example of this is Aspergillus niger. A young colony, say, of 48 hours old is yellow. At 5 days old, it's black. Black as anything. So if you say on the phone, I've got a cup to here of Aspergillus niger, and they go, is it black? And you go, no, it's yellow. Oh, it can't be then, because his is a really old one, yours is a young one. Age of the colony is super important. They change colour a lot as to age. So media was worth a mark, and so was age. If somehow in your long explanation you half-linked size to age, I gave you a mark. But if you just said size, it doesn't really work by itself. You said growth rate maybe in size. That's kind of linking back to how old the colony is. Then things that can cause onychomycosis that are not dermatophytes. If you wrote in that answer trichophyton or eflocosum or mycosporum, which are dermatophytes, so what do we know that are not dermatophytes that cause onychomycosis? Acremonium, Strophylaeropsis, Candida albicans, Fusarium, Boswellidium, possibly Aspergillus. There's probably a few more that I've not thought about. With your spelling, I was reasonably generous because these are all new names for you. As long as your name made some reasonable sense, I was okay with it. This one here I don't like that much because when you go Aspergillus, it's almost always a contaminant. To prove that's causing onychomycosis requires lots of reculturing and showing the same thing over and over again. Those ones there were the ones that would, to me, would be nice, simple to pick. And then question 9. In microbiotic agar, you have a broad spectrum antibacterial. It's usually chlorphenicol or it might be gentamicin. They're the two most commonly used. As long as you said either of those drugs and said antibacterial, that's worth a mark. And then the other drug is cyclohexamide, and it's anti-saprophyte and pro-dermatophyte. As long as you said that was there as an anti-saprophyte drug, that's okay. So add up your marks, put them in a big circle on the front of your page at the top, and I'll put them in the computer. On your way out, hand it back to me, and I'll see you next week. One more reminder about the case studies. Find one this week, really, get onto it, and then let me know.